mitochondria, there are many folds that leave space for the protein complexes in the intermembrane. Oxidative phosphorylation is a metabolic pathway that uses energy released by the oxidation of nutrients to produce ATP. The electron transport chain are electrons that are transported to meet up with oxygen from respiration at the end of the chain. In the beginning, NAD and FAD, which are reduced coenzymes, are transporting through the electron transport chain, and it continues to break down the original glucose in order to create ATP. In protein complex 1, the dropping off of electrons will happen. While it will be losing hydrogens, it will also be passed to ubiquinone, to complex 3, to cytochrome C, and lastly to protein complex 4. As seen on the diagram, the cytochrome C is located in between protein complex 3 and 4, but there are other cytochrome complexes that are embedded in the phospholipid bilayer. In protein complex 4, it passes this electron that was dropped off at complex 1 to a molecule of oxygen, which is a highly electronegative. Then the oxygen atom will pick up 2 hydrogens which are floating in the matrix to release water. As you can see that FADH2 isn't being dropped off at complex 1 but at complex 2. But the problem with that is since the drop-off is taking place in complex 2, which means we're not using complex 1, and we're not using complex 1, we'll miss out on the hydrogens, when missing out on hydrogens, it means there's less potential to create that proton motive force. Through this process, the hydrogens want to move back into the matrix through the ATP synthase.